right, next up for us is the lymphatic system. So the lymphatic system is really your immune system. And so that's what we're going to be focusing on, is how does your body respond to the threats around it? How does it control the microorganisms or microinvaders that get into your system? All right, so in a nutshell, the lymphatic system transports excess body fluids away from the interstitial spaces in most tissues. It returns proteins to the bloodstream, and it helps defend the body against infection by disease-causing agents or pathogens. So when we look at this, the lymphatic system transports excess body fluids away from the interstitial spaces. So the interstitial spaces is really the extracellular space. It's just the space that we find in between the cells. We've got fluid in between those cells, and we're going to move that fluid into our lymphatic system is what we're going to try to do. So we've got four primary functions: to drain excess interstitial or extracellular fluid, to return any proteins to the bloodstream so we can redistribute them accordingly, to transport dietary lipids, and to carry out immune responses. I don't know why that has two twos there. Let me fix that real quick. There, boom. All right, so when we get into this, we need to understand immunity. Immunity is defined as the ability to ward off damage or disease. Susceptibility is a lack of resistance. So right out of the gate, we need to know the difference between immunity and susceptibility because they're not the same thing. With our immune system, we've got two basic types of immune system. We have our innate immunity. This is non-specific. This is the stuff that you're born with. The defenses that are present at birth, and it provides a rapid response to all types of disease. Adaptive immunity or specific immunity, this develops in response to contact with specific pathogens, disease-producing microbes. It occurs more slowly but can remember previous contact with specific pathogens. Boom. All right, so the big difference is innate immunity is the immunity that you're born with, like your skin classifies as part of your innate immunity. It's the stuff that you're born with. The adaptive immunity is the immunity that you learn. You get exposed to a cold, you develop the antibodies to fight that cold if you ever come across it again. So that's the difference between innate immunity and adaptive immunity. So our interstitial / extracellular fluid is the fluid that surrounds each of the cells of our body. Once the fluid passes into a lymphatic vessel, we call it lymph. So there's no difference in the actual chemistry or makeup of our lymph and our interstitial or extracellular fluid. It's just the location that's different. So it's the same fluid, just in a different location. Once it moves into a vessel, we refer to it as lymph. Before it's in that vessel, we call it interstitial or extracellular fluid. And we can see in our picture down on the bottom right how we've got the space in between all those cells and how lymph moves into a lymphatic vessel and then starts its journey. So good little picture, just kind of showing you an overview of most of the lymphatic system there. On the right hand side, all of the little vessels and stuff, how it comes in and helps drain all the excess fluid. All right, so lymph forms from extra or excess interstitial fluid, and your body's going to make about 3 liters a day. So every day you make about 3 liters of lymph. Lymphatic capillaries start the lymphatic drainage. They have a greater permeability than our blood capillaries, and they are one-way structures, much like our veins are one-way structures. So fluid can flow in, but not out. Lacteals, specialized lymphatic capillaries that we find in the small intestines to absorb dietary lipids. I didn't have a good place to stick lacteals in there, but I wanted to make sure that we had that. So there it is. So our lymphatic capillaries we compare with our vessels a lot because they travel with our vessels. We often see we'll have an artery and a vein traveling together, but we also have a lymphatic capillary traveling with them. And so that's one of the reasons we make a comparison between our lymphatic capillaries and our vessels. Our lymphatic trunks and ducts. So we look at the trunks first. So our lymphatic vessels in a particular region of the body all merge together to form a bigger lymphatic trunk. So it's just a bigger vessel on its way back. All All lymphatic vessels flow into one of the following trunks. We have our lumbar trunk, which is going to drain all of the lymph and the lymphatic vessels from your lower limbs and your pelvis. The intestinal trunk is going to drain the lymph from the stomach and guts. Bronchomediastinal trunk is going to drain the lymph from the thoracic wall, lung, and heart, so basically your chest. The subclavian trunks drain lymph from the upper limbs, so there's your arms. And then the jugular trunks are going to drain lymph from the head and neck. So the big thing here is we have a left and right lumbar trunk. We have a left and right bronchomediastinal, left and right subclavian, left and right jugular, but we only have one intestinal trunk. Everything else comes in pairs, not the intestinal trunk. There's only one of those. Alright, so lymph passes from all of these trunks into two channels. So all of those trunks, there's 9 of them all together. They're all going to dump into one of 2 ducts. The first is the thoracic duct, and it's the big one. This is the main duct for returning lymph to the bloodstream. It starts out as a dilation called the cisterna chyli, and it's going to receive the lymph from your right and left lumbar trunk, the intestinal trunk, lymph from the left jugular, left subclavian, and left bronchomediastinal trunk. So that means the thoracic duct drains lymph from the left side of the head, the neck, and the chest, the left arm, and the entire body below the ribs. So it's doing almost everything. And then our thoracic duct is going to empty back into our venous bloodstream at the junction of our left internal jugular vein and our left subclavian vein. So where those 2 veins come together and merge, boom, that's where our thoracic duct is going to dump all this lymph it's collected back into our bloodstream. Our other duct is the right lymphatic duct, and it's a little one. It's a little one. It's only about 1.2 centimeters long. It receives lymph from the right jugular, the right subclavian, and the right bronchomediastinal trunk. So it gets the right side of your head, your right arm, and the right side of your chest. But that's it. Everything else goes into the thoracic duct. The right lymphatic duct empties into the venous bloodstream at the junction of the right internal jugular and the right subclavian. So it's the same thing as our thoracic duct, but we're just on the right side because it's the right lymphatic duct. So this next picture, we already talked about the picture on the left a little bit, just showing all of the lymphatic vessels. The picture on the right now is what I want to point out. So everything in green on this right side is what gets drained. I'm sorry, on this left side is what gets drained by the thoracic duct. So we can see how much of your body the thoracic duct is actually draining, and everything in this yellowy, orange, peachy color is what gets drained by the right lymphatic duct. So we can see it's not an even workload. Things are not distributed evenly. The thoracic duct is definitely doing the lion's share of the work when it comes to dumping lymph back into the venous bloodstream. One second. All right. So back to our picture. We can see at the bottom down here how the lymphatic vessels, or the lymphatic ducts, I should say, how they dump their lymph back into the venous bloodstream. All right, moving on. Another little picture just showing how everything flows together. There we go. So we start with our lymph out there floating in between the cells. That lymph gets picked up by the lymphatic capillaries. Those lymphatic capillaries are going to enter a afferent lymphatic vessel, which is then going to pass through a lymph node, and it's going to leave that lymph node through an efferent lymphatic vessel. Then it's going to go into one of the lymphatic trunks, just depending on what part of your body it's in. Then those lymphatic trunks are going to drain into one of the 2 collecting ducts, and those 2 collecting ducts are going to dump into the internal jugular and subclavian vein junction on the left and right side, just depending on which collecting duct it is that we were talking about. All right, so the flow of lymph works by the same mechanisms that return venous blood to the heart. So we're also using our skeletal muscle pump that we talked about in our vessels chapter. When the muscle contracts, it squeezes the blood up through that next valve so it gets a little bit closer to the heart. Same thing with our lymphatic vessels. When it compresses that vein and pushes the blood, it's also compressing that lymphatic vessel and pushing the lymph up through the next valve. Respiratory pump, working with the diaphragm again to help get it over that last little bit. And here at the bottom, lymphatic vessels contain valves, just like our veins, to ensure that one-way flow of movement. So some more pictures just showing you how lymphatic vessels are spread throughout the body. I don't think I use any pictures on the test for our lymphatic system. We have enough pictures with the heart and the vessels already. So I don't think there's a practical component to this, but I'll keep you updated. All right, lymphatic organs and tissues. We have primary lymphatic organs that are going to include our red bone marrow and our thymus. Red bone marrow and our thymus. So there's only two. So we have two primary lymphatic organs and tissues. And these guys are the site of stem cell division and development of immunocompetent B and T cells. So immunocompetent means these are the cells that are just capable of mounting an immune response. So we got our B and T cells with our red bone marrow and our thymus accordingly. So let's look at the red bone marrow first. Our red bone marrow has stem cells that make both our B and our T lymphocytes. So it's making the B cells and it makes the T cells, the red bone marrow. But this is where our B cells mature. Our T cells take off and head out. Our B cells stick around in our bone marrow, and that's where they mature. Hence, B for bone. B cells, B for bone. Now, our T cells, they head over to the thymus. Thymus, of course, is that bilobed organ. Our immature T cells migrate out of the red bone marrow and to the thymus to multiply and mature. We get some dendritic cells derived from monocytes that aid in the maturation process of these T cells. And then we get epithelial cells, super important, because these guys help educate our immature T cells to distinguish self from foreign molecules, meaning our epithelial cells help teach these T cells not to attack our own body. We're looking for stuff that doesn't match to go after. Only about 2% of our immature T cells will actually make it through the maturation process. The rest die by apoptosis. All right, our secondary lymphatic organs and tissues are the site of most of our immune responses. With this, we get our lymph nodes, we get our spleen, and we get our lymphatic follicles. So with our lymph nodes, we get about 600 bean-shaped lymph nodes spread throughout our body. Typically, they're located along the lymphatic vessels because we need to funnel the lymph through them. They often appear in groups. We got lots of afferent vessels leading into the lymph nodes, and then just one or two leading out. So there's lots of ways into a lymph node, but there's only one or two ways out. And in that way, we can direct the lymph out one of these two vessels and make it go through the entire node for the cleaning purposes. So we got one efferent vessel leaves at a hilum or depression. So a capsule covers and extends into the node as trabeculae, but this helps divide the node into various compartments that the lymph has to go through. So within the lymph node, lymph's going to enter the sinuses, a series of channels that contain branching reticular fibers with lymphocytes and macrophages embedded in them. So lymph enters the node through one of these afferent vessels. The first thing it's going to roll into is the subscapular sinus, which is part of the pathway, going around the edge. Then it's going to go into the trabeculae sinus, and finally the medullary sinus in the middle there before it leaves out one of these efferent vessels at the hilum of our lymph node, which is just that little depression in the lymph node. So this functions as a type of filter for foreign substances. We get macrophages that destroy by phagocytosis and lymphocytes that destroy by the immune response. So I'm going to back up one slide here. So all along this pathway that this lymph gets pushed through and has to go through each lymph node, all the way through it, all along this pathway, The whole thing is just embedded with lymphocytes and macrophages. And they're there just to clean out anything that's not supposed to be there. Anything that triggers them or sets them off, they're going to try to eat it, get rid of it, and destroy it. Then once it makes it all the way through the lymph node, that lymph gets passed to the next lymph node. And the whole process continues all the way through the lymphatic system to make sure all of that stuff is clean before we dump it back into our bloodstream. All right, the spleen. Spleen is the largest mass of lymphatic tissue, located on the left between the stomach and the diaphragm. We got two types of tissue in our spleen. We got white pulp, which is the site of B and T cell immune responses. We got a red pulp. Red pulp has three different functions. It works to the removal of worn out or defective blood cells and platelets. We can use it for platelet storage, and we can do it works to produce blood cells during fetal life. So our pictures on the bottom just show the location of the spleen and how it works to help clean out the blood, take out busted up blood cells and platelets, store platelets, all of that stuff. Next up, we have our lymphatic follicles. Concentrations of lymphatic tissue without a capsule, just where we've got loose lymphatic tissue kind of hanging out. We refer to this as mucosa-associated lymphatic tissue, or MALT tissue, scattered throughout several tracts of several organs, and they carry out immune responses by intercepting pathogens that enter the body from passageways that open to the exterior. So that's the key thing about MALT tissue is any spot in your body where there's an orifice, where there's an opening to the outside world, right inside of that opening, inside of your body, we're going to have some MALT tissue to help stop anything that we don't want getting in from getting in and getting into our system. We use the tonsils as an example here. We can see the tonsils at the back side of the mouth here. There are little rounded bulges on each side. Tonsils strategically placed to participate in immune responses that get ingested or inhaled. We have 5 tonsils. We got a single pharyngeal tonsil, also known as the adenoid. We find that in the back wall of our nasopharynx. We got 2 palatine tonsils on the posterior region of your oral cavity, so right inside your mouth at the back. These are the 2 that they take out when they do a tonsillectomy. And then we got some lingual tonsils, 2 lingual tonsils at the base of the tongue. So here's a little picture showing you where those tonsils fall into place for ingested or inhaled pathogens. All right, now we have to distinguish between innate versus adaptive immunity in a little bit more detail. So we're going to start with innate immunity. Innate immunity is non-specific, and we say this just because it acts against all microbes in the same way. Doesn't matter what it is, if it triggers your innate immunity, it's going to react to it in the exact same way as it does everything else. Our first line of defense of innate immunity, this is our external defense. So first line of defense is external, provides a physical and chemical barrier, and with this we're talking about your skin and your mucous membranes, the things that stop things from getting into our system right out of the gate. So our skin and our mucous membranes are part of that first line of defense of innate immunity. Skin, closely packed keratinized epithelial cells, and then with our mucous membranes, they trap any invaders or microbes in the cilia, sweep them away. Coughing, sneezing, swallowing, all part of that first line of defense of innate immunity. Chemicals and fluids with tears, saliva, urine, defecation, vomiting, help expel microbes, and acidity discourage bacterial growth. The second line of defense of innate immunity. So the first line of the defense was your skin and your mucous membranes. Our second line of defense now is internal, but this is still non-specific innate immunity. So if something gets past your skin or mucous membranes and triggers your second line of defense in innate immunity, it's still going to react the same way to everything else. We get 4 categories here. The first thing is our natural killer cells. They attack any body cells that display abnormal membrane proteins. So we talked about glycoproteins and how they determine blood type. Specifically, with our natural killer cells, they're looking at any membrane proteins that they don't recognize, and they're going to go after it if they find something new or different. Natural killer cells bind to infected cells and kill in two ways. They can either release perforin, which create channels, also known as perforations or holes in the plasma membrane. This allows our extracellular fluid inside that cell to, the extracellular fluid to leak into that cell and causes the cell to burst. We refer to this as cytolysis. Cytolysis. Or granzymes. This causes the infected cell to self-destruct, aka apoptosis. So with the natural killer cells, we're looking for perforin and we're looking for granzymes. All right, our next component in the second line of defense with innate immunity is phagocytes. And as their name suggests, they're going to perform phagocytosis. They're going to reach out, they're going to grab something, they're going to put it into a vesicle, and they're going to pull it into their body, and they're going to break it down and kill it. So phagocytosis occurs in 5 stages. With phagocytes, we're talking about primarily our neutrophils and our macrophages for the most part. So 5 stages to phagocytosis. The first is chemotaxis, where we get a chemical attraction for phagocytes. So something has triggered a phagocyte to roll in and take control. The second stage is adherence, where it's going to come up and attach itself to these foreign particles. Then it's going to ingest them. It's going to pull the particle into itself inside of a vesicle. Next up is digestion. The vesicles get sent to a lysosome, which is then going to help break it down and destroy it. And lastly is the killing phase, where our lysosomal enzymes kill and digest that particle. So we need to know the 5 stages of phagocytosis in order, because it happens in that order. So another picture just showing you all how the 5 stages work their way through, starting with the microbe and chemotaxis at the top left, and then it makes its way all the way through the phagocyte here. Our third category is inflammation. This is the body's attempt to slow the spread of foreign particles. So inflammation is a natural, normal process. It's your body trying to take control of things, get a situation under control. 4 characteristic signs of inflammation: redness, swelling, heat, and pain. Lastly is a fever. Hypothalamus sets an abnormally high systemic temperature. This helps inhibit microbe growth. It intensifies the effects of interferons, and it speeds up the body's reaction. Everything moves faster at a higher temp. Body's reactions included in that. And lastly, we have our antimicrobial proteins. Antimicrobial proteins. These discourage microbial growth. Most important of these are the interferons. So that's really what I want you to get. Interferon or antimicrobial proteins equals interferons. So our interferons are proteins produced by cells that get infected by a virus. Once produced, they move to uninfected cells and stimulate antiviral proteins to help block viral replication. So our first cell in our picture down here, boom, gets invaded by a virus. That cell then sends out the interferons to all the surrounding cells to help slow down or block that viral replication process, so that virus is limited on its ability to spread. So antimicrobial proteins, interferons. And that is innate immunity. Now, adaptive immunity, now this is your learned immunity. This is specific. So the body's defenses against each particular antigen. So antigens, substances recognized as foreign that produce an immune response. We're talking microbes, pollen, anything that's not supposed to be there. This is the distinction from innate immunity, is that we are looking for something specific for a particular foreign antigen. We have a memory of previously encountered antigens, so a second exposure prompts a more rapid and vigorous response because we've already been there once. So when it happens again, we're more prepared. So with adaptive immunity, we're talking about the maturations of our B and T cells. Both lymphocytes arise from stem cells in the red bone marrow, but they get their names from where they mature. T for thymus, B for bone. Remember, both of these B and T cells come from the bone marrow, but we name them for where they mature. So B cells mature in the bone marrow, T cells mature in the thymus. And that's really the big distinction, is because we're going to talk next about what happens with our T cells and then what happens with our B cells. And so we need to make that distinction because they act differently. Antigens. The term antigen derives from its function as an antibody generator. The epitope is a small part of an antigen that is recognized as foreign and triggers an immune response. Human immune system can recognize at least 1 billion different epitopes, and as a rule, our antigens are foreign substances. Major histocompatibility complex molecules, or MHC molecules, so it's not so much of a mouthful. These are just molecules that we find inside of our cells. They have self-antigens on all of our cell membranes, and they help our T cells distinguish our own body cells that are supposed to be there from the foreign cells that are not. When we get an autoimmune disorder, that's when our T cells start attacking our own body cells. Antigen presenting cells. This is a special class of cells that process and present antigens, strategically located where our antigens are likely to penetrate our innate defenses. After processing, they migrate to lymphatic tissue to present to the T cells and trigger an immune response. So what that means between our MHC molecules and our APC cells is we've got a cell here. So let me read through this. Processing of an endogenous antigen, where we get a fragment of the antigen bind to the MHC inserted into the cell membrane, signals as an infected cell for an immune response. So we've got a phagocyte here that's coming along. It's like, hey, what is this? Let's eat it. And so it's got this little purpley antigen thing outside of the cell. So it reaches around and it grabs it, pulls it into the cell, and it's like, oh yeah, we don't recognize this. There's something going on. Then it will take the broken up pieces of that endogenous antigen, or that it's got now inside of the body, and it will give a piece of that to an MHC molecule. Then the MHC molecule will move it to the outside or to the edge to the cell membrane, and it will stick it out and then go show it to a piece of lymphatic tissue to trigger that immune response. So we can say, hey, there's something fishy going on here. So the antigen presenting cells, the entire cell that goes and shows a piece of an antigen to some lymphatic tissue, where our MHC molecules are just a part of that we stick into the membrane to hold that antigen in place until we get to that lymphatic tissue. So that's really the big difference between the MHC molecules and the antigen presenting cells. All right, adaptive immunity. So this is kind of the Rosetta Stone for this PowerPoint. If you can understand this slide, everything else is easy, because it really repeats itself for the most part, with just a few minor differences between each. So clonal selection. This is the process by which B or T cells divide and differentiate into a clone of a cell that can recognize the same specific antigen as the original activated cell. We get two major types of cell from a clone. We get effector cells that are named so because they have an effect. They actually go and do something. And in this case, they're going to carry out an immune response to destroy or inactivate antigen-bearing substances. Or that clone can become a memory cell that doesn't participate at all, but it can remember if the same antigen is encountered again in the future. So that's really the clonal selection process. We take either a B cell or a T cell, something triggers it, some sort of response triggers it, and then it can either become, it's going to divide, and it can either become an effector cell or a memory cell. An effector cell can actually go do something about it, or a memory cell where it remembers it for future reference, and we kind of hold on to that information. That is the clonal selection process. We're starting with one cell that's going to split into either an effector cell or a memory cell. So Types of adaptive immunity, we got two types here. Cell-mediated immunity, this is cytotoxic T cells directly attack invading antigens. This is effective against pathogens, cancer cells, or transplant tissue. And it always involves cells attacking cells. So cell-mediated immunity, there's a couple things here. When you think of cell-mediated immunity, you should be thinking of T cells. T cells are cell-mediated immunity, and this involves cells attacking other cells. Antibody-mediated immunity, we're talking about the B cells. And our B cells are a little different because they turn into plasma cells, and then our plasma cells synthesize and secrete a specific antibody to bind to a specific antigen. So we got lots of specificity going on here. And this is mainly effective against viruses, bacteria, fungi, and often both of these responses are working together. So cell-mediated, we're thinking T cells. Antibody-mediated, we're thinking B cells. So the word antibody has a B in it, so antibody-mediated B cells. All right, so let's talk about these in more detail. So if we get into our cell-mediated, again, we're thinking about our T cells here. This begins when receptors on a T cell recognize and bind to a specific foreign antigen fragment. This binding is the first signal for the T cell activation, but a second signal, co-stimulation, is still required. So we use cytokines, specifically interleukin-2 or IL-2. So cytokines, IL-2, interleukin-2 provide the necessary co-stimulation. And we require this co-stimulation in our body to prevent an immune response from occurring accidentally. We don't want cells attacking cells accidentally inside our own body. So we require these cytokines, specifically interleukin-2, to give co-stimulation to say, yes, we should definitely activate a T cell, or no, we should not go ahead and activate these T cells, just depending on the circumstance. So during T cell development, we can get two major types of cells that exit the thymus. So we get helper T cells and we get cytotoxic T cells. So if we look at our helper T cells first, once activated, our helper T cells, they're going to undergo that clonal selection, and then they have a choice. They can become an active helper T cell, which is going to be our effector cell here doing something, or it can become a memory helper T cell, which is not active, but it's going to remember for later on. So if our activated helper T cell becomes an active helper T cell, then it's going to start secreting its cytokines, because it's really here just to help. And to help, we're going to need those cytokines. We're going to need that co-stimulation to get the rest of the T cells going. So these cytokines start secreting their interleukin-2, needed for almost all immune responses, and is the prime trigger for T cell production. So that is helper T cell clonal selection. Helper T cells are going to become active helper T cells, and they'll start secreting their cytokines. Memory helper T cells are not active, but they're going to remember for later. If we come across this again, we're going to be ready to go. All right, our other type of T cells are the cytotoxic T cells. So just from the name alone, cyto means cell, toxic means death. So these are the killing cells, essentially. So cytotoxic T cells recognize antigens on the surface of the body cells. We get co-stimulation occurring from the cytokines, the interleukin-2 produced by the helper T cells. So now we're ready to go because we've got those cytokines going. Once activated, our cytotoxic T cell clonal selection will produce either active cytotoxic T cells, which now go and attack the cell that's infected with the antigen. Or we're going to become a memory cytotoxic T cell, where it's not active, but it's going to remember for next time, and when we need it, we're going to be faster. So that's cytotoxic T cell clonal selection. So if we kind of summarize everything here, we get our T cells that get activated, and they're going to become either helper T cells or cytotoxic T cells. Helper will become active helper to start secreting interleukin-2 with the cytokines. Cytotoxic, if it becomes active, is going to become an active cytotoxic T cell and directly attack a cell. Versus our memory helper and memory cytotoxic T cells are not active, but they remember for next time. So elimination of invaders with our cytotoxic T cells. So cytotoxic means killing cells. They recognize and attack target cells and deliver a lethal hit. They act a lot like the natural killer cells do, but cytotoxic T cells have receptors for a specific molecule. So there's a big difference right there. Our natural killer cells, remember, they are non-specific, so they act towards all invaders the same exact way. With our cytotoxic T cells, we're looking for a specific molecule to go after, something specific to trigger that specific cytotoxic T cell. And our cytotoxic T cells kill by, like the natural killer cells, they use granzymes to trigger apoptosis. They use perforin to punch holes in the cell membrane of a target cell. But then they also use granulysin to destroy intercellular microbes, or they could use lymphotoxin to fragment a target cell's DNA. Without the DNA, that target cell is not doing anything. So while cytotoxic T cells are acting, they also secrete substances that attract phagocytes to the area, and those phagocytes are going to come in and help clean up any mess that was made by our cytotoxic T cells. Antibody-mediated immune responses. Remember, with antibody-mediated, we're talking our B cells now. So our B cells are different in that they stay within our lymphatic organs and tissues. We're not going to chance losing our B cells because they are precious. So we keep them safe inside of our organs and tissues. In the presence of a foreign antigen, a specific B cell forms a plasma cell that secretes a specific antibody to disable the antigens. And B cells can respond to an unprocessed antigen if it's close enough to something that they've already processed before. So what we're basically saying with this is where T cells go out and directly attack their target cells, B cells don't. B cells turn into plasma cells. Plasma cells then secrete their antibodies, and those antibodies are what actually go out and do the fighting for them. And that way we're able to keep the B cells safe because we got somebody else, specifically these antibodies, doing the fighting for us against a particular target cell. So with our B cells bind to an antigen, our helper T cells have provided the co-stimulation with their cytokines and their interleukin-2 that we need. Once activated, our B cell clonal selection will either turn into plasma cells, which are the effector cells here, or our memory B cells, which are like everything else memory. It's not active but can become active if we need it to. It's going to remember. Our plasma cells then start secreting specific antibodies. Now, something that's important to note here, all of the B cell clones are only capable of secreting one type of antibody, and that is determined by whatever the original B cell was secreting. So if, say, you get like type A flu, for instance, and you start making B cells here, your B cells are going to turn into the plasma cells, and they're going to secrete specific antibodies that can only go after type A flu. But it's only the type A B cells that will be activated in the first place. With our antibodies, 5 classes of antibodies. We got IgG. This is the most abundant one, and it's the only one that actually crosses the placenta. We got IgA to help protect our mucous membranes against bacteria and viruses. We got IgM, which is the first to be secreted after initial exposure. IgD we find on the surface of B cells, and these guys are involved in B cell activation. And then IgE, involved in allergic or hypersensitivity reactions and against parasitic worms. Now, our antibodies work in various ways. They can produce a neutralized antigen by blocking the attachment of the antigen to body cells. They just don't ever let that antigen in. They can use the immobilized bacteria to limit the spread to nearby tissues. They can agglutinate antigens, causing antigens to clump together, making them easy pickings for phagocytosis. Or they can enhance phagocytosis and act like a flag to bring in more phagocytes to go after the antigens to begin with. Lastly, immunological memory. Due to long-lasting antibodies and very long-lived memory cells, your body creates its own unique immunological memory. And this is what becomes the basis for immunizations and vaccines. So there is the lymphatic system. If you have any questions on this, shoot me an email. Otherwise, good luck.